In this video, we will be explaining the process of ubiquitination and proteasomal degradation. Proteasomal degradation is one pathway used by cells to break abnormal and other targeted proteins down to amino acids and peptides. The mechanism by which proteins are targeted for proteasomal degradation is through the ubiquitin conjugation mechanism. Ubiquitination involves the process of a small ubiquitin protein covalently attaching to substrate molecules to form a polyubiquitin chain. This process requires the involvement of three enzymes. E1, or the ubiquitin activating protein, where the ubiquitin becomes activated in an ATP-dependent manner. The ubiquitin is then transferred to E2, or the ubiquitin conjugating enzyme, which combines with our third enzyme, E3, or ubiquitin ligase. This last enzyme facilitates the transfer of ubiquitin from E2 to a lysine residue in the substrate protein, generating a ubiquitinated substrate. This entire process repeats until the chain is built and the protein can be recognized for degradation. This is a proteasome which consists of a core and a regulatory particle. The cylindrical core of the proteasome has 4 stacked 7-member rings, which each have 7 distinct polypeptides. The central chamber is lined with proteolytic sites. 2 rings of the alpha-type subunit form antechambers on each end of this central chamber, and are responsible for restricting access into the proteolytic chamber. The regulatory particle varies based on the function of the proteasome. There are 10 lid proteins that bind ubiquitin on the protein, and deubiquitinating enzymes remove the ubiquitin from the protein. This removal is crucial because it allows the protein to fit into the proteasome. The 10 subunit base includes an ATPase that unfolds the protein and threads it through the central channel. When an abnormal protein is recognized in the cell, it quickly gets ubiquitinated. The 3 ubiquitin enzymes are at work here. E1 is activating the ubiquitin before being transferred to E2, which combines with E3, allowing the ubiquitin to be transferred onto a lysine residue on the protein. This is repeated to form a chain of ubiquitin molecules that will be recognized by the regulatory particle on the proteasome. The lid of the proteasome is where the ubiquitin gets removed from the protein. Now that the protein has no ubiquitin on it, the ATPase can unfold the protein and open up the gate for the protein to move into the antechamber. Inside, the antechamber recognizes the protein and opens up, allowing the protein to move into the central core. As the protein moves through the central core, specific beta subunits cleave the protein and break it down into amino acids that can be recycled by the cell.